In biology, kingdom prokaryote Monera. Today we will discuss about bacterial cell and its structural components. As first of all, picture is given of bacteria. Bacterial cell structure. Flagella and their functions. Flagella helps in motion or movement of bacteria. They are extremely thin hair-like appendages. External outgrowths which grow or come out of the cell are called appendages. They come out through the cell wall, begin or originate from basal bodies. Now the picture of prokaryotic cell. They have flagella, cell wall, cell membrane, capsule. Basal body is a structure just beneath the cell membrane in the cytoplasm. And in flagella, the protein present is flagellin. On the basis of presence of flagella, patterns of arrangement of flagella, their numbers, they are classified into different groups, taxonomic groups. Now, what is taxonomy? It is the branch of biology that deals with classification of organisms and evolutionary relationships among them. As it is given, taxonomy is the study of classification of organisms and evolutionary changing relationships among them on the basis of evolution. Taxonomic groups of bacteria on the basis of flagella, like these are Atrichus, Monotrichus, Amphitrichus, and Peritrichus. Pictures are given. Monotrichus, they have only one flagella. Now we will discuss them one by one. Atrichus, A means not having. Bacteria are without any flagella. Monotrichus, mono means one. Bacteria have single flagellum that is present at one pole. Next is Amphitrichus. Bacteria have tuft, cluster of flagella at each of the two poles. Lophotrichus, if a tuft or cluster of flagella is present only at one pole of bacteria, then it's called Lophotrichus flagella. And Peritrichus, in this form, flagella surrounds the whole bacterial cell, as in the picture. Most of the bacilli, that is rod-shaped bacteria and spiral-shaped bacteria, have flagella. But cocci, which are rounded or spherical, they rarely have flagella. Basic functions of flagella. Primary function is to help in motility, movement of bacteria. And bacteria having flagella are called flagellated bacteria. They detect and move in response to some chemical signals. This type of behavior shown by bacteria to the chemical signals is called chemotaxis. Chemo for chemicals and taxis is for their movement. Next are some structures which come out of the cell surface, and these are called pili and their functions. Their definition is pili are hollow, non-helical, non-spiral, filament-like appendages. Pili are smaller than flagella. They are not involved in motility or movements of bacteria. True pili means well-developed pili, and they are present only on gram-negative bacteria. Gram-negative bacteria are stained with pink color. Pili are made up of a special protein, pyelin. They are primarily involved in mating reproduction process between the cells. This process is called conjugation, in which exchange of genetic material takes place. Some pili function as mean of attachment of bacteria to different kind of surfaces. In their structure, next is cell envelope, which is for protection. It is the outer wrapping of bacteria, covering of bacteria. Bacterial surface and walls are very diverse, that is different. Collectively, complexes of layer external to the cell living substance protoplasm is called cell envelope. That is, it is the protective layer. It includes capsule, slime, and cell wall. One by one, you will learn about it. Capsule. Bacteria produces capsule which is made up of repeating carbohydrate polysaccharide units, proteins, or they are made up of both proteins or carbohydrates. Capsule is tightly bounded to the cell. It has thicker, gummy, sticky nature, and it gives sticky characteristics to bacterial colonies of covered in a capsule encapsulated bacteria. Next structure is slime. Some bacteria are covered with loose soluble sheet of large macromolecules called slime capsule. Function of slime is to give greater pathogenicity of bacteria. Definition of pathogenicity is property of microorganisms like bacteria to cause diseases. Slime protects bacteria against phagocytosis, that is, they are not being swallowed, eating process of a cell. Next, the most striking and characteristic feature of bacteria is cell wall. Beneath the extracellular substances external to the cytoplasmic membrane, cell wall is present. It is hard, rigid structure and it determines the shape of bacterial cell. Its function is to protect the cells from osmotic lysis. Osmotic lysis means entry of excess water in a cell. Then a cell wall in plants actually prevents the membrane from bursting. That is, plant cell doesn't burst because it has additional protective layer, cell wall. But in case of animal cells, as they lack cell wall, so they may burst due to entry of excess or extra water. Cell wall is only absent in bacteria mycoplasmas. Sir Hans Christian Gram was a scientist and he developed the technique of Gram staining. According to this, bacteria could be divided into two groups based on their response to Gram stains. These are gram-positive bacteria and gram-negative bacteria. Number one is gram-positive bacteria. They are stained purple. They retain the color of primary dye due to the formation of crystal violet CB1 complex. That is, they are basically purple stained. And second are gram-negative bacteria. They are colored or stained pink and they retain the secondary dye color, which is pink. And in diagram it is mentioned, gram-positive bacteria stained purple and gram-negative bacteria are stained pink. Next, as you know, bacterial cell wall is made up of a special molecule which is called peptidoglycan. Next, there are ribosomes, DNA, plasma membrane. The cell walls of most bacteria have unique macromolecule peptidoglycan and its amount varies in different types of bacteria. It is composed of a framework of long glycan chains. Glycan chains are actually made up of sugar molecules. They are cross-linked or interlinked, that is connected with each other with peptide or protein fragments. The intact or complete cell wall has chemical constituents like sugar molecules, teichoic acid. Now, diagram of bacterial cell, that it's flagella, pili, ribosomes, these are present. There are some conjugated molecules like lipoproteins, lipids and proteins, lipopolysaccharides, lipids and carbohydrate polysaccharides. And they are linked to the basic component of cell wall, peptidoglycan. Several bacterial groups lack cell wall structure, characteristic of gram-positive or gram-negative bacteria. Some bacteria have no cell wall at all. Cell wall of ancient, old group of bacteria, that is archibacteria, they are different from eubacteria, true bacteria. They don't contain any peptidoglycan in their cell wall. Their cell walls are composed of other materials like proteins, conjugated molecules like glycoproteins, carbohydrate plus proteins, and polysaccharides. Glycan chains, it means large carbohydrates with sugar molecules joined chemically. Next is cell membrane or plasma membrane. Just beneath the cell wall is cell membrane. It is very flexible, thin, and it surrounds the cytoplasm. Plasma membrane is very delicate in nature. That is, any damage to the plasma membrane results in death of an organism. Bacterial membrane differs from eukaryotic membranes in lacking cholesterol, sterols. Cell membrane regulates the transport of various substances like proteins, sugars, nutrients, metabolites. Metabolites are the substances that take part in metabolism chemical reactions. Cell membrane also contain enzymes for respiration or regulation of respiratory processes metabolism. There comes cytoplasmic matrix, that is space in the cytoplasm. It is the substance lying between the plasma membrane and nucleoid. Matrix is largely composed of water. 70% of bacterial cell is made up of water. Bacteria lack a true cytoskeleton. Plasma membrane and everything within it is called living substance, protoplast. Cytoplasmic matrix is a major part of protoplast. In cytoplasmic matrix of bacteria, large, discrete, prominent, distinct structures are present, which are chromatin or nuclear body, ribosomes, mesosomes, granules, and nucleoid. What is nucleoid? A bacterial cell. Unlike the cells of eukaryotic organism, they lack discrete, distinct chromosomes and nuclear membrane. As you know, in prokaryotes, no nucleus is present. This nuclear material or DNA in bacterial cell occupies a position near to the center of the cell, as in picture it was mentioned. This nuclear material is single, circular, double-stranded DNA molecule. It aggregates or collects as an irregular shaped dense area in bacteria called as nucleoid. Chromatin body, which are thread-like structures in an extremely long molecule of DNA, and is tightly folded and fits inside the cell space or components. Bacteria have single chromosome, so they are haploid. There are so many names of nucleoid like nuclear body, chromatin body, and nuclear region. Nucleoid is visible in light microscope just after staining with a stain called Fulgen stain. E. coli or Escherichia coli bacteria closed circle chromosome measures approximately 14,000 micrometers. There are also some other structures in bacteria, just as spores, plasmids, ribosomes, mesosomes, cysts, granules, storage bodies. Now we will discuss them one by one. Number one are plasmids. Many bacteria contain plasmids in addition to chromosomes. What are plasmids? These are circular double-stranded DNA molecules. They are self-replicating but are not essential for bacterial growth and chemical reaction metabolism. Plasmids contain drug-resistant, heavy metals, disease and insect-resistant genes on them. That is, they totally get protected. Plasmids are important vectors or carriers in modern techniques like modern genetic engineering. As we can say that plasmids make copies very quickly. So important genes can be inserted in them and we can get copies from the plasmids. Next are ribosomes. As you know, they are involved in protein making. Ribosomes are composed of RNA and proteins. They are loosely attached to the plasma membrane. They are actually protein factories. There are thousands of ribosomes in healthy growing cells, but they are smaller than eukaryotic ribosome. As we discussed that prokaryotic ribosomes are 70S and eukaryotic ribosomes are 80S. Next, there are some extensions which come out from the cell membrane. They are called mesosomes. Cell membrane invaginates, fold inward into the cytoplasm and forms a structure called mesosomes. They are involved in making copies of DNA called DNA replication and cell division. And some mesosomes are involved in exports of enzymes, exocellular enzymes. Enzymes are also involved in respiration and they are present on mesosomes. Next are some granules and storage bodies. Bacteria exist in competitive environment. It means that nutrients are in short supply over there. Nutrients are not available. So they tend to store extra nutrients like glycogen, sulfur, fats, phosphates. Cell also contains some waste materials which are then excreted or removed out from the body. as common waste materials are alcohol, lactic acid, and acetic acid. Bacterial spores are called endospores. They make it either external to the vegetative spores called exospores or within the vegetative cells called endospores. They are metabolically dormant or inactive bodies, and they are produced at the later stage of cell growth. Now, spores are resistant to harsh physical environmental conditions like light, high temperature, desiccation, dryness, shortage of water, pH, and chemical agents. Under favorable conditions, they grow, germinate, and they form special type of cells called vegetative cells. Finally, we will discuss about cysts. They are dormant, inactive, thick-walled, dryness-resistant structures, and they develop during specialization of vegetative cells. They can grow under suitable conditions whenever light, temperature, they are suitable. They are not heat resistant, that is, they can't resist excessive heat. And finally, structure of bacterial cell is given.